Everyone. I would like to thank Dr. Sarma for a nice introduction. Also, I'd like to thank the organizer for inviting me to present our work here. And my work is going to be a little different from what has been, like presented and discussed. It's more on system biology. And this work was done when I was in the Department of Chemical and Biomolecular Engineering, National University of Singapore. Like most of the students there were chemical engineers. I was the only biologist, so I was helping with the biological part, and they were doing the mathematical part. So my talk is plant system biology and how that can be applied to rice, also any other plant, to understand the metabolic and regulatory characteristics under different abiotic and biotic stress conditions. We all know rice is consumed worldwide. Also, it's produced worldwide. And since Green Revolution, there is really, like a lot of increase in rice production. But then the question is how we are going to feed the growing population. There is prediction that by 2050, there will be 9 billion people. And even if the rice production is increasing, it's the production is affected by different biotic and abiotic stresses. Like we have drought stress, flooding stress, salt stress, light stress, also heat stress, mineral stress, and biotic stress. So it's very important to characterize the stress response mechanism to improve the productivity of rice, also other crop plants. And we use the system biology approach. And we know now many plants have been sequenced. Rice was the second plant that was sequenced in 2005. We have rice genome. Also, we used to have many microarray data earlier. Now we have proteomics data. Now we have metabolomic data. Also, we have transcriptomic data. Then what we are going to do with all these data? We can how to combine these data to get the knowledge. And that can be done through system biology approach. And to use the system biology approach, there are different types of modeling which you can do for the plant. The first one is the interaction-based. This is a very simple model where all the pathways are connected to each other, like the cake pathway, like only you look at the pathways, but you cannot simulate any condition. And the second one is constraint-based, where you can do, like simulate the flux through different pathways. And the third one is mechanism-based. That is most comprehensive, but then it lacks required experimental parameters. So normally people use the constraint-based modeling to develop the model. And this is a very old picture. In 2011, if you look, these are many models have been developed initially for the lower microorganism, bacteria, and things like that. For plant, there are only very few, like Arabidopsis, Brassicanthaphus, and Geomates. But now many plants, the genome scale metabolic model for many plants have been developed. So what we did when I was in chemical engineering, we wanted to develop a genome scale metabolic model of rice. Before that, what we did, we developed a core model, and it is like tissue specific. We developed a model for core model for leaf specific rice model, another one seed specific rice model, because you can only study the leaf tissue here. Here you can only study the seed tissue. And to develop that, you have to follow some manual methods. The first one is draft reconstruction, where you compile genes, reactions, and pathways information from different databases. Now, these days, many plant databases are available, like cake, Brenda, Gramine, PMN, many, Riceic, Araceic, many pathways. So we first collected the pathway genes and the reactions. Next one was manual refinement, which really takes a lot of time. And many of our undergraduate students, they helped us in curating these pathways, where you have to, in plant, you have to localize the specific reaction to the specific organelle. Suppose mitochondria should have the specific reactions, chloroplast should have, that's the plant is a complex, like it's a complex structure. So we need to have the localization information, then also construct a gene-protein reaction and remove dead ends. If there is dead ends, then the flux may not be able to flow through, and also close the gaps. So it took a lot of time, manually curating all this. Then finally check the reaction and balanceability, because many reactions are reversible, some are unidirectional. That also you have to make sure. And then in the third step, once all these are done, third step is model evaluation and simulation. How can you be sure that your model is working? It's similar, more similar to the plant system. So we evaluate based on the experimental data. And for these core models, we had only 3 organelles like cytosol, plastid, mitochondria, and only the primary metabolic pathways like carbohydrate metabolism, photosynthesis, fat metabolism, amino acid metabolism, but no secondary metabolic pathways. So this is the validation we did for the leaf model. What we did, we look at the difference in photorespiring leaf cells under normal and drought stress condition. And when we simulate, we found that when there is decrease in CO2 uptake, the cell growth cell growth and both CO2 and cell growth both decrease when there is increase in drought stress. And the next simulation, what we did, when we increase the flux through photorespiratory pathways, we found when we simulate the drought condition, we found there was increase in flux through photorespiratory pathway and decrease in TCA cycle enzymes. This is the TCA cycle enzyme. This is the higher flux through the photorespiratory pathways. And for the seed-specific model, what we did, we grow rice culture cells in different sucrose, glucose medium. And we found that our experimental data quite matching to the simulation data. And when we look at we again look at the metabolic difference of germinating seeds in air and anoxia. As we know that when the rice seeds germinate, they elongate coleoptile, because normally we grow rice during rainy season and the fields are normally flooded. So the coleoptile elongate, reach the surface of the water, and then they get oxygen. After that, the root and shoot initiates. And rice is the only plant that can germinate under hypoxic and anoxic condition. And we all know that during anaerobic process, mainly they produce energy through the glycolysis and ethanolic fermentation. And when we simulated our rice seed metabolic model, we found that, ethanolic fermentation, there was an increase in flux through ethanol synthesis, and the TCA cycle was truncated. It was not, there was no flux going through the TCA cycle. And there was also a possible role of GABA, because GABA is also quite important during the abiotic stress condition in maintaining pH, and also helps in transport of metabolites from organelle to organelle. And then for the leaf-specific model, we did more analysis that we did through distribution of the essential genes across rice central metabolism in photorespiring leaf under normal condition and also drought stress condition. And we found the red ones are the essential genes, blue ones sub-essential, and the green, these are the non-essential genes. What we found that again, that most of the lethal enzymes of the photorespiratory metabolism are found to be associated with Kelvin cycle, Glas-Gillett cycle, and many enzymes of the TCA cycle. So after doing this, we are quite confident that our model is working, and it's quite, giving the, it's matching to the experimental data, and whatever is known, it's giving such type of data. Then once we are confident, we wanted to do some analysis that is metabolic and transcriptional regulatory mechanism that is associated with anoxic adaptation of rice coleoptile. And for this, we use some publicly available data. And we did the flux analysis, and our simulated flux, this is the experimental data, and they were also quite matching. And same here, we found the flux was much higher through the ethanolic fermentation. And what we did to look at the transcriptional regulation of the anaerobic seed germination in rice, we did the promoter analysis by taking the important genes like sucrose synthase, pyruvate decarboxylase, alcohol dehydrogenase, and aldehyde dehydrogenase. We did the promoter analysis and we found the common binding site for ERF, transcription factor, B, zinc finger, and BGIP. And when we look at the expression of those genes, we found there is good correlation between these transcription factors. And we hypothesized that there could be, because of these transcription factor, MIB, BGIP, ERF, and zinc finger, they could be controlling the transcriptional regulation of the coleoptile elongation in rice. Then next, in the meantime, we develop from the core model to rice genome scale network, where all the pathways are connected, but we cannot do the flux analysis. So to utilize our network, we did more analysis. We wanted to identify the candidate network hubs. Those are involved in metabolic adjustment of rice under drought stress by integrating transcriptomic data and genome scale metabolic network. This is the flow chart of our analysis. Here also we use some public data. That is drought stress transcriptome of rice, different tissues like roots, leaves, and panicles at different stages like tillering, butting, and panicle elongation stage. So here what we did, because we cannot have the flux, the genome scale metabolic model was not ready, but it was a network. So what we did, we took the transcriptomic data, that is thousands of data, we mapped to our model. And then we separated out all metabolic genes. So we were only with the metabolic genes. Then we divided them to different clusters depending on the tissue and different growth stage. These are the metabolic processes associated with these metabolic genes. Those were identified by mapping to the rice genome scale network. Then we did promoter analysis and then look at the expression of the genes. Then we hypothesized that those could be the potential transcription factors involved in metabolic adjustment of drought tolerance in rice in different tissues and different conditions. So these are the metabolic pathways for different, like panicle butting stage, root and panicle elongation stage, so different stages. And then these are the pathways, some it was upregulated, some downregulated. And then we did the promoter analysis for different cluster of the genes. And we found the binding site for many transcription factors like MIB, BGIP, ERF, RAB1, ABI4, AP2ERF, DAP. So these are the patterns we found for different clusters. If you look at this, there is a mixture of transcription factor binding site. But in one cluster, it was really interesting. Only MIB, the binding site for MIB transcription factor. So depending on the situation, the transcriptional regulation also varies. And that you can see from this figure, how the pattern changes. Then we look at the expression of those transcription factor genes in different clusters. And it was interesting that the transcription factors we identified are already known in rice because drought stress in rice has been extensively studied. And many transcription factor, the new genes that is identified have already been, the field trials have already been done. So then it gave us more confident that, okay, like already many are known and whatever we identified, it's matching so well. So in summary, these are already known, and through our mapping to our metabolic network, we also identified the same binding site, transcription factor binding sites like all these transcription factor. And our result also shows that ABA plays a key role during the metabolic adjustment in rice during drought stress. And there are also two signaling pathways operates, like ABA dependent and ABA independent. And that shows that shows through this, you still can identify the regulatory mechanism that controls the spatiotemporal gene expression pattern and global metabolic fluxes. So our ultimate goal is to identify candidate genes that is relevant to the metabolic changes for targeted selection in rice breeding. So we believe that this method can be applied to any other, of course, stress condition to identify the candidate genes and any other plant. This is the overall summary figure of our work. Many are known, many are not known. Of course, these needs experimental validation. And then in the meantime, our genome scale metabolic model was ready, where we can do the flux analysis. So for that, to test our model, we did some work on light-specific metabolic and regulatory signature of rice. This is the genome scale metabolic model. Here we have more reactions, like 2,164 open reading frame. And then we have more compartments, like we have chloroplastid, mitochondrion, endoplasmic reticulum, peroxisome, vacuole. It's the complete cell. And with the gene protein reaction and most of the metabolic pathways, whatever we could find from the different databases. And then again, we validate the model through the experimental data, and it was quite nicely matching with the experimental data. This is it's more or less same, like what we did for the drought stress. And here, what we did, our collaborators in Korea, they did this experiment. Rice seedlings, 2 weeks old seedlings, they were grown in blue, different LED light, blue, green, red, and dark. If you look at the phenotype of the plants, you can see the obvious differences. In blue, they are shorter with wider and darker and thicker leaves. In red, they are more elongated and thinner leaves. Dark, of course, no chlorophyll, so yellow leaves. And in green, again, elongated leaves. And they also did some secondary metabolites analysis. They found that in blue light, the secondary metabolites like beta-carotene, alpha-carotene, zeaxanthin, and lutein all higher compared to red light. And also they did phenolic compounds measurement. It's the same thing. The blue light, it was much more compared to red and also white. So when we analyzed the data, what we found that gene expression under dark is quite different from the other light condition. And then among the light treatment, blue and red, they have diverse pattern. Blue is here, red is here. There is no difference between white and green. And then we look at the expression of individual metabolic pathways. Again, we map to our model, and then we separate out the metabolic genes and look at the pathways. And here the white is used as the control. And the most of the pathways regulated in blue, like photosynthesis, Kelvin cycle, gibberellin metabolism, terpenoid biosynthesis, and most of the pathways down-regulated in red. And in the dark, of course, carbohydrate metabolism, amino acid metabolism, and fatty acid degradation all up-regulated. And photosynthesis, flavonoid metabolism, all down-regulated. Then next, what we did, we took the 20 reporter metabolites for each condition, and we did the promoter analysis. And based on to look at the transcriptional regulation of these rice seedling growth under different light conditions, And here you can see the blue is for the blue light and red for the red light, and the black is the common light signal. There is a major difference between the transcriptional regulation under the blue light and red. We found many BG transcription factors. Those are already known related to photosynthesis, stomatal opening, and chloroplast movements. And we also found many new transcription factors. Then again, they need experimental validation. And if you look at the red, there are number of MIPs, but we didn't find many of these transcription factors. Those were like activated during the blue light condition. And the growth here, the height of the plants are much shorter compared to the seedlings grown under the red light. That could be, the ABA could be inhibiting ethylene and down regulating the length of the hypocotyl. So this is the summary of our work. Our results shows that constraint-based modeling can be used as an effective tool to investigate plant cellular metabolism. And when we integrated the metabolomics and the transcriptomics, you can, you can look at the rice cellular behavior. And the blue light has highest metabolic efficiency, but not plant growth means in terms of height. And these transcription factor, BHLS and BGIF, they are blue light specific, and also some of them are already known. And the MIB is more red light specific. So it shows that this kind of work can be potentially applied for crop improvement, designing a better growth environment, and synthetic circuit design, and also, of course, many more things. So by the time we finished our rice metabolic model, after that, many other, now I think many metabolic models, genome scale metabolic models have been available for other plants like soybean, gia maize, tomato, also few more. And the same approach can be applied to other cereals. And then the next part is the biotic stress. We wanted to also develop a genome scale metabolic model for Xanthomonas oryzae that causes the rice leaf blight disease. And we all know that the yield loss is severe. It's 20 to 30% in moderately affected areas, and it can be up to 50% in severely affected area. Of course, that can be controlled by chemicals, but then it's strain dependent. So it's we wanted to also study, through system biology approach to see, whether we can identify something. Since many omics data available these days, we compared the genomic study on 3 different exo strains. And we found that there are several gene clusters representing various functions like virulence, hypersensitive reaction, pathogenicity, and the regulation of pathogenicity factor, that is RPA, also the exapolysaccharide production, that is GUM, and extracellular plant cell wall degradation. And also similarly, different metabolomics data is also available, that is key metabolites closely related to virulence. So, but even if these data are available, we don't know how they interact. To study that, we wanted to develop a genome scale model for exo. And we did several experimental, we developed the model like similarly we developed for the rice. And we did some batch culture experiment to validate our model. We found that the growth is more or less same to the experimental data. And the model can successfully simulate the growth from several carbon sources like glucose, sucrose, fructose, cellulose, xylose, mannose, lactose, starch, and many things. And the predicted values were highly consistent with the experimental measurements. And in the model, we have these pathways like the exocentral metabolism, diffusible extracellular factor, diffusible signaling factor, that is the quorum sensing, then the xanthan biosynthesis, xylan and cellulose degradation, polyhydroxybutyrate biosynthesis. And then we wanted, once we are sure that, okay, our model, we can do the flux analysis after experimentally validating the model. As we all know, when you apply nitrogen fertilizer, then the disease aggravates. So to simulate that, what we did, we simulate the flux in nitrogen sufficient and nitrogen limited condition. In nitrogen sufficient condition, we provided on the ammonia uptake by unconstraining agent, the continuous flow of ammonia. And in the limited condition, we limited the ammonia uptake to 50%. And we found that there was upregulation of gluconeogenesis, glycogen biosynthesis, and degradation. And we found lower flux within the carbon catabolic pathways, of course, higher flux through those pathways. So it shows that the exo might divert the resource towards storage carbon biosynthesis, such as glycogen, and subsequently degrade them during nutrient starvation. Because we all know they colonize in the xylem, and xylem is normally poor in nutrients. So they could be reserving those storage carbon for the further use. The prediction of the upregulated gluconeogenetic reaction, that is phosphoenol pyruvate synthase reaction, is in good agreement with experiments for Xanthomonas oryzae. So then we hypothesized that the disruption of such pathways in exo could be a key to reduce the severity of blight during the application of nitrogen fertilizer. But then, of course, we need experimental validation. And we found that then what we did, we used some publicly available microarray data of exo wild type strain and the mutant RPFF mutant. And what we found that when we did the simulation, we found that the highest fold change was found in the reaction catalyzed by DSF synthase encoded by RPFF gene. Xanthan biosynthesis was significantly downregulated. Okay, sorry, I think the slide is not here. And then starch sucrose metabolism, cell envelope biosynthesis, oxidative phosphorylation, biotin metabolism, all were downregulated. Porphyrin and chlorophyll metabolism and citrate cycle were upregulated. And we also found upregulation of xanthan B2 that is involved in the synthesis of a diffusible factor that is in the mutant, and it suggests a possible inverse relationship between DSF and DF signaling. So we hypothesized that this could be a strategy employed by EXO to conserve metabolic resources due to the overlapping role of DSF and DF signaling, and that indicates the degree of complexity of regulatory process in EXO pathogenesis. And then next, we wanted to predict the antimicrobial properties in EXO. That we did through gene essentiality workflow, like we did the lethal gene analysis for photorespiring leaves. And what we found that in the B, these are the metabolic pathway genes essential for growth, and these are for the invasion. And here you can see the DSF, DF, and xanthan, and the genes associated to those are more. And this stage is the virulence, and here even it's more. Our analysis shows that the growth, there are 24% genes, those found to be essential, out of the total genes. Some of the major players are involved in invasion and virulence. Those include xanthan, DSF, and DF. And amino acid, cell membrane, and virulence factor biosynthetic pathways are the major proportion of the essential genes. So these essential genes represent the theoretical anti-blight targets that, of course, additional knowledge, we need to have additional knowledge about that possible virulence properties. And that would maybe in the future help us to narrow down to more promising targets in controlling the disease. I think that's the end of my talk. We have, okay, that's the summary. I already explained constant-based modeling can be used as an effective tool to investigate cellular metabolism in XO. And we could integrate the omics data to find more information. And these are the pathways, they are key central metabolic pathways potentially contributed to the XO virulence. Then integration of relevant transcriptomic data revealed that many metabolic routes through which the virulence factors are, especially through DSF-DA, that could contribute to exopathogenesis. Finally, using this systematic analysis involving gene essentiality and right host homology search, we propose experimental testable antibacterial targets that could be crucial in controlling the disease. And this one I'm not going to explain. We did some model for lettuce. Lettuce growing in fluorescent light and LED light, and this is combination of green and blue. And when we experimentally validated, we could see the difference in both the our model validation, and lot of difference. And these lettuce under blue and red, they have a lot of secondary metabolites like anthocyanin and other things. For this work, many people were involved for the modeling and the simulation, for the experimental validation. So I would like to thank the National University of Singapore, also A-Star Singapore, and also the Korea, like we had the grant from Next Generation BioGreen program, and we had a lot of Korean collaborators, and they helped us to do the experiments. And these are the people involved in this work, and many students, PhD students, final year students, they helped in manual uration, and the simulation was done mainly by the chemical engineering students. I'm a biologist, like not expert in mathematics, but they were handling those part. I was handling the biology part. And currently I'm working in US Environmental Research Institute. Here we work more on plant microbe interaction, and I'm not going to show those work here.